Hello, this is Dr. James Camp at Lee College in Baytown, Texas, and we are about to take a trip inside the ovaries. The ovary is the last of the bean-shaped organs that we'll cover in A&P2, so of course it's going to have certain parts. Uh, the germinal epithelium on the outside of the ovary here takes the place of what we call the capsule in most other bean-shaped organs. Then there is an ovarian cortex that contains the ovarian follicles. Okay. And there's an ovarian medulla here just in the middle that contains blood vessels, lymphatic vessels, nerves, things that sort of feed and inform the ovary. And then finally, there is the dimple in the bean, a hilum through which vessels and nerves enter and exit. The ovary, of course, is the site of oogenesis, the production of egg cells. Oogenesis follows the same gametogenesis pathway that we looked at in the male on the last lecture. So the oogonium undergoes mitosis to produce primary oocytes, which undergo meiosis 1 to produce secondary oocytes, which undergo meiosis 2 to produce an ovum. Now, you may remember that I said that the female contribution to conception is an oocyte, but here I have the last cell being an ovum. What's up with that? The secondary oocyte is actually what is ovulated from the ovary. It is then only completes meiosis 2 to become an ovum at the moment of conception. So it waits until the last possible minute to complete meiosis. Okay. To briefly review, the timing of male gametogenesis is really, really simple. In utero, the testes develop, but there's no spermatogenesis. During childhood, that is ages 1 to 10 or so, there's a pause in reproductive development. The boy just grows in a more or less reproductively neutral way, and there's still no spermatogenesis. Beginning at puberty, which we used to place at about age 13 for males, but it seems to be getting earlier in some populations, the testes are fully active. So every single day, spermatogonia divide to produce more spermatogonia and to produce primary spermatocytes. The next day, those spermatocytes divide to become secondary spermatocytes. And the next day, those divide to become immature spermatozoa. Actually, to become spermatids, which then become immature spermatozoa. And then those go on through a long, drawn-out process in the epididymis to an organ called the epididymis to become mature spermatozoa. Finally, those mature spermatozoa are forwarded through the male reproductive system, which we'll talk about next time, and they are ejaculated if they're lucky, into a female reproductive system where they can fertilize an egg. But the point is, all of these stages, spermatogonia to primary, secondary to spermatids, etc., all of those steps happen every single day in a male, beginning at the time of puberty and ending pretty much upon death. Okay, the timing of female gametogenesis is way different. Ovaries develop in utero, and prior to birth, all of the oogonia that are going to become oocytes go through that transition. So they go through mitosis to become primary oocytes. So it can be said, it's been said that a girl has all the eggs she's ever going to have when she's born. What you mean by that, I guess, is, or what is meant by that, is that a girl has all of the primary oocytes she's ever going to have when she's born. Again, during childhood, that first 10 to 12 years of life, there's a pause in reproductive development, and so there's a similar pause in oogenesis. All of these primary oocytes are still there, but they're not doing anything. Beginning at puberty, which again we used to place at about 12 for girls, but it seems to be getting younger as well. Primary oocytes, once per month, a primary oocyte is activated to become a secondary oocyte, and that secondary oocyte is ejected from the ovary through the process called ovulation. Unlike the male, where ejaculation is actually ejecting the sperm from the body, ovulation is simply moving the oocyte from one compartment in the ovary to another compartment in the uterine tubes. Only upon contact with sperm does the secondary oocyte complete meiosis to become an ovum. And since contact with sperm has already happened, that ovum immediately then goes on to merge with the sperm to form a fertilized zygote. I like to make the comparison here that when I was dating the woman who is now my wife, she used to not get completely ready for a date until she could actually see me walking up the sidewalk to her house to pick her up. At that point, she would finish getting her, I don't know, her makeup on or her shoes or whatever it was that she hadn't gotten ready. Just didn't want to waste the effort until she knew that I was there for the date. And the oocyte seems to have the same philosophy, not going to waste the effort finishing meiosis until we know that the sperm is there to complete fertilization. Okay. Oogenesis begins in utero as the ovaries of a female fetus begin to produce egg stem cells known as oogonia. This is just summarizing what we just talked about. Most oogonia degenerate, but just before birth, a portion develop into primary oocytes, which are slightly larger cells. Biologists often use this abbreviation 1 with a degree to mean primary, 2 with a degree to mean secondary. Okay, beginning at puberty, 1 oocyte each month in most women. Some women double ovulate and are more likely to have twins for that reason. 1 oocyte each month becomes a secondary oocyte as it is ovulated from the ovary. Strange as it sounds, each oocyte only completes meiosis 2 to become an ovum, a completely meiosised cell at the moment of fertilization. We're now going to take a little detour away from oogenesis to talk about folliculogenesis. During development, each oocyte is surrounded by a protective shell called a follicle. You may remember the word follicle from when we studied the thyroid, that the thyroid gland had all of these circular things with pink in the middle that were called follicles. Similarly, the ovary has all of these circular structures with an oocyte in the middle, with an oocyte here in the middle, and just as with the spherical structures of the thyroid, the spherical structures of the oogenesis, the spherical structures of the ovary are called follicles, and they participate in endocrine activity as well. Okay, so let's meet the follicles. The follicle family contains 4 major growth stages. At the time of birth, a girl's ovaries are entirely made of these primordial follicles. And what sets a primordial follicle apart is that the oocyte is surrounded by simple squamous cells known as follicular cells. Okay, and at this point, those cells serve only a protective function. They are not active in an endocrine fashion in any way. As the ovary makes a transition from primordial to primary follicle, these cells become more like the endocrine jumble that we've seen before in other endocrine cells, sort of a semi-cuboidal cell, but not really a formal cube. It's just, a bunch of circular cells jumbled together. These cells are now called granulosa cells. You can see why they get that name. They look kind of granular. While the follicular cells are purely for protection, the granulosa cells over here begin endocrine activity. They secrete estrogen. Okay, so at this point, when a primordial follicle is activated to become a primary follicle, we start getting secretion of estrogen out into the body. Okay. The primary follicle develops into a secondary follicle. The main thing that we see in a secondary follicle is the development of this open space where there is fluid instead of cells, and that open space is called an antrum. Okay, so secondary follicles are also sometimes called antral follicles because they have an antrum. They continue to have granulosa cells, which continue to secrete, in this case, even more estrogen. Okay, then finally the secondary becomes a tertiary, which is also known as a mature follicle because it is almost ready to be ovulated. It's also known as a vesicular follicle because it's got this big antrum that looks like a vesicle, okay? And it's known as a Graafian follicle after a Dr. de Graaf who discovered them in the first place or characterized them. So here we have some interesting features that I'll highlight in just a minute when we get into the microscopes. But once again, we still have granulosa cells that are secreting gobs of estrogen at this point. But we have our oocyte now somewhat isolated from the rest of the follicle. I call it the egg on a stalk design. And at some point, this mature follicle is able to rip apart at this point, and this entire package right here gets ovulated. Okay, so let's compare how gametogenesis stacks up compared to folliculogenesis. Remember, we said that the oogonia become primary oocytes in utero just before birth. Okay. Then there's a break during childhood. And then puberty happens. And once puberty happens, all of these things happen for the rest of the woman's reproductive life, which is from about age 12 to about age 50. So the first thing that happens, so when the girl is born, her primary oocytes are all surrounded by primordial follicles. Okay. At the beginning of each month, a primordial follicle is activated to become a primary follicle. Over the course of the first week of development, that primary follicle further develops into a secondary follicle, so it develops an antrum, remember, continues to get bigger, produce more and more estrogen. During the second week, the secondary follicle develops to become a tertiary or mature follicle. And at the end of the second week, the mature follicle ruptures. Okay, which is where the secondary oocyte is then ovulated. Okay, if fertilization occurs, the secondary oocyte converts to an ovum and then immediately to a zygote. And then implantation occurs. We have all sorts of other developments that happen after that. No follicle around the developing embryo at this point. The ruptured follicle closes in on itself and becomes something called a corpus luteum, which literally means a yellow body, which produces a different set of hormones than what the follicle was producing. And eventually the corpus luteum, if a baby doesn't take hold, if a pregnancy doesn't take place, degenerates into a corpus albicans, from albus, white, so a white body, which is mostly scar tissue, or pretty much scar tissue, and will eventually degenerate. And that is the ovarian cycle. Each month, one primordial becomes a primary, one primary becomes a secondary, one secondary becomes a tertiary, one tertiary or mature follicle ruptures for ovulation, and one ruptured follicle becomes a corpus luteum. And then if the cycle is to continue, because we don't have a pregnancy, the corpus luteum becomes a corpus albicans, and we start all over with a new primordial follicle. Okay, so let's move over to the microscopes here. You're welcome to come into lab and look at this for yourself. In fact, I encourage you to. This is what the ovary looks like at the minimum magnification. It says 4x objective here. I think this was actually a picture taken under 2x magnification, just so that you can see the entire follicle. I mean the entire ovary, sorry. Notice the hilum here, the kind of light lavender medulla here, and the deep purple cortex that is most of the ovary, filled with follicles. There's a big follicle, there's a small follicle. Here are some teeny tiny follicles. If you're following along on your microscope, you want to find a section just deep to the germinal epithelium. So note there's the germinal epithelium right along there. Just deep to that, you'll see some little tiny cells that almost look kind of like soap bubbles on the edge of a film of soapy water. We're going to zoom in on those and go to the next slide. Okay. The small circles here, these are the primordial follicles. Okay. The bigger circle over here, in case you hadn't guessed, that is these are primordial. This bigger circle over here, that is a primary. Okay. Can you tell a difference between the small circles and the larger one? Well, hopefully, of course you can. Note that the oocyte, the internal part, is about the same size. The oocyte maybe gets a little bit bigger as it goes to being a primary follicle. The main difference, though, is these cells around the follicle. They are simple squamous. We have a simple squamous cell border that makes this a primordial follicle. Okay, whereas here we have what I'm going to call a cuboidal-ish wall. Some of these cells end up being fairly tall. Some of them end up being kind of flat, but the average cell is kind of cuboidal. These are the granulosa cells. Okay, so these are follicular cells. Here around the simple squamous, these are granulosa cells around the primary. The other thing you'll see is the very first hint here of a zona pellucida, that protective jelly around the oocyte. We zoom back out to our 10x objective and scan around, and we'll find a large irregular oval. Here's the oval. with an island in the middle. That's your oocyte. There's your zona pellucida. Okay, it's now a fully developed zona pellucida. Can you tell two different tissues in this? Hopefully what you'll notice is the kind of endocrine jumble of granulosa cells here compared to the very ordered cells here right around the oocyte, and there's kind of a boundary between those two tissues there. This tissue here is called the corona radiata, the round crown, and it's going to the corona radiata is going to be ovulated with the oocyte, and it's going to remain as a protective coating around the oocyte as we move on, or to protect it as it moves through the female reproductive tract. All right, so that's the end of our part one on sort of the microanatomy of the ovaries. Please take a break, look over your notes, and then continue with part two on the microphysiology, the hormones and cycles of the ovary.